Hi everyone, my name is Max Sabalis, and today I will be talking about the paper Raman Spectroscopy Reveals New Insights into the Zonal Organization of Native and Tissue Engineered Articular Cartilage, published by the Burkholt et al. Group. In the study, the authors used confocal Raman spectroscopic imaging to uncover greater structural and biochemical complexity in articular cartilage than the traditional three-zone model suggests, and they also apply these insights to evaluating tissue-engineered constructs that aim to mimic the native tissue's depth-dependent features. So let me start with some context. Articular cartilage is the smooth, specialized tissue lining the ends of bones in diarthrodial joints, such as the knee and hip. It is classically portrayed as having three distinct zones: the superficial zone, with collagen fibers oriented parallel to the articular surface, the middle zone, with somewhat random fiber orientation, and the deep zone, where collagen fibers align perpendicularly. These differences in orientation, combined with biochemical variations in components like glycosaminoglycans, which are often abbreviated as GAGs, and water, help articular cartilage distribute loads and maintain low friction. However, conventional techniques like histology with dyes or biochemical assays of proteoglycan content often provide only a snapshot or approximate quantification, potentially missing more subtle gradations across the thickness. Raman spectroscopy, which detects inelastic scattering of light at vibrational frequencies specific to different molecular bonds, it offers a far more detailed analysis that does not require staining or destructive sample preparation. This possibility prompted the authors to investigate whether additional zonal layers might exist beyond the familiar three-zone framework. So what were the authors trying to find out? In essence, the authors wanted to answer whether confocal Raman spectroscopy could identify biochemical and structural nuances that have been overlooked by conventional methods. Their guiding hypothesis was that a label-free micrometer resolution method would reveal depth-dependent changes in critical peaks like the 1410 band, which is associated with GAG, the 1245 amide 3 band, which reflects collagen, and the 1668 amide 1 peak, which is also primarily associated with collagen, but also sensitive to orientation. They also wondered if these data, when subjected to multivariate analyses, such as k-means clustering, could uncover multiple sublayers within what we typically call the superficial, middle, and deep zones. Furthermore, the authors wanted to find out whether these refined insights might be applied to engineered cartilage by comparing how its composition evolves over time relative to that of mature native tissue. To address these questions, they collected two main categories of samples. First, they took full-depth osteochondral plugs from adult bovine joints, removing slices that span from the articular surface down to the calcified cartilage boundary. Second, they created scaffold-free cartilage constructs by culturing bovine chondrocytes for 2, 4, or 6 weeks, allowing the cells to deposit their own extracellular matrix. They then performed confocal Raman spectroscopy with a 532 nanometer excitation laser, scanning large regions on the order of 1,000 times 400 micrometers at roughly 1 to 2 micrometer resolution, and so that each map contained thousands of spectra. Polarized Raman signals were used to detect collagen fiber orientation changes through depth, particularly by examining ratios such as 1668 to 943 inverse centimeters, which shifts as collagen fibers move from parallel to perpendicular. To incorporate biomechanics, they also conducted nano indentation tests to measure the local elastic modulus, E, and hydraulic permeability, which is represented by K, at different depths. Finally, they analyzed the spectra with methods like multivariate curve resolution, principal component analysis, and k-means clustering to identify underlying biochemical components and group spectrally similar regions. Figure 4 is one of the central highlights of the paper because it visually demonstrates how k-means clustering of the Raman data produced at least 6 distinct zones in the native articular cartilage rather than the traditional 3. In the figure, each color on the clustered map corresponds to a unique cluster of spectra that share characteristic shapes and intensities in key Raman peaks. For instance, the collagen amide 3 signal at 1245, the GAG signal at around 1410, and even the water and apatite contributions can differ from cluster to cluster. The authors noted that the intensity of the 1668 band, which is sensitive to collagen fiber alignment, changes more than once between the surface and the calcified boundary, indicating extra subzones in what was formerly lumped into the superficial or deep regions. Crucially, these variations were not random noise, but rather consistent, repeating patterns across several replicates, and this suggests that the tissue has multiple fine layers with unique orientations and relative concentrations of ECM molecules. Figure 5 then links these sublayers to mechanical measurements via nano-indentation. The authors performed indentations at different depths and plotted how the elastic modulus, E, and hydraulic permeability, K, changed as they moved deeper from the surface towards the tide mark. On the graph, the modulus increases substantially with depth, hitting its highest point closer to the calcified interface, while permeability decreased in the same region. This difference implies that water is more easily squeezed out near the superficial zone than in the deeper one, and that the deeper zone must be stiffer to serve as a robust transition region before calcified cartilage. Increasingly, the paper points out that these mechanical patterns did not correlate simply with total collagen or total GAG concentration, as measured by the 1245 or 1410 band intensities. Instead, alignment and microstructural features, which we can particularly infer from the 1668 amide 1 orientation dependence, appear to have a bigger impact on how stiff or permeable each sublayer is. This finding reinforces the idea that microarchitecture goes hand in hand with mechanical function. Turning to Figure 7, we see how the authors leveraged the Raman-based approach to assess tissue-engineered constructs over time. The graphs displayed a zonal quality metric that integrates the discrepancy between native and engineered cartilage in terms of GAG, collagen, and water distributions across the tissue thickness. Specifically, they track how intensities at 1410, which is GAG, and 1668, which is collagen, they track how they evolve from the surface region down to where the PTFE membrane meets the engineered tissue. At 2 weeks, the engineered construct's GAG distribution diverges significantly from that of native cartilage. However, by 6 weeks, the GAG pattern begins to resemble the native tissue more closely. On the other hand, collagen remains mismatched compared to the robust collagen peak and orientation near the 1668 in native samples. And this discrepancy suggests that reproducing the water and GAG distributions is easier during early culture, while replicating the complex fibrillar architecture of collagen in deeper zones is likely the greater challenge, requiring more time or specific mechanical and biomechanical stimuli. There are a couple key implications that arise from these results for both basic cartilage biology and regenerative strategies. First, identifying 6 or more sublayers in a single cross-section forces us to think more carefully about how each region contributes to shock absorption and shear resistance, rather than assuming one large middle zone. Second, the mechanical data confirms these sublayers have functional significance, meaning future therapies to repair or engineer cartilage must aim to replicate that fine-grained structure. The authors particularly emphasize how the extra transitions in collagen alignment near the tide mark might serve as an anchoring or load dampening mechanism that is crucial for long-term cartilage health. Finally, the direct comparison of native to engineered tissues with a quantitative measure of zonal fidelity highlights that even if total biochemical content is similar, the spatial arrangement still needs to match the natural complexity for effective mechanical performance over time. The main strengths in this study include its label-free nature, since Raman spectroscopy doesn't require exogenous stains or destructive sample prep, and its high spatial resolution. This allows the authors to compile rich data sets that capture both composition and fiber orientation. Additionally, coupling these findings with nanoindentation provides a functional tie-in that helps interpret why these new sublayers matter for biomechanical integrity. Nevertheless, there are a few limitations. The author's mapping approach, although it was rigorous, is time intensive, which may pose challenges for clinical translation or high throughput screening. Similarly, the paper examines samples from a limited number of animals, leaving open the possibility of inter-individual variation that could slightly alter the sublayer thickness or compositions. And while the tissue engineered constructs do begin to show improved gag deposition, the alignment of collagen proved more elusive, hinting that the protocols might need further optimization. Now, looking forward, the findings suggest multiple avenues for expanding this research. Longer-term cultures with mechanical stimulation or growth factor supplementation may lead to engineered tissues that better encapsulate the deeper collagen arrangement found in mature cartilage. Further exploration of the orientation-sensitive Raman peaks could yield more quantitative definitions of each sublayer, allowing researchers to design more precise scaffolds, potentially with gradient porosity or anisotropic fiber alignments, to match complexities uncovered here. Another promising direction involves applying this approach to disease models such as osteoarthritis to see exactly how the newly identified sublayers might degrade under pathological conditions. Finally, if faster Raman mapping protocols can be developed, one might envision a clinical application in which surgeons or pathologists access repaired cartilage directly within the body, ensuring that the multi-zonal organization is truly restored. Finally, these are the sources that I used for this presentation. Thank you for listening.